Hello, SC12. This is Paul Whitford from Northeastern University. I'm going to tell you a little bit about how we're using the Massachusetts Green High Performance Computing Center to learn about the dynamics of biomolecular assemblies. In our group, we focus on large-scale assemblies that have to, in biology, that have to come together to execute common biological roles. This first example is called the ribosome. In the ribosome is composed of about 60 molecules, and it produces proteins in the cell. And what I'm going to show you first are just the two endpoint configurations of one of the essential conformational changes called translocation. And what we do is we construct all atom models that have hundreds of thousands of atoms to simulate spontaneous transitions from one state to the next. Now, while each of these simulations is not enormous on an HPC scale, in order to say anything meaningful about biological dynamics, you need to simulate hundreds, if not thousands, of events so you can talk about the statistical properties of a system. In this case, what we're looking at is a large motion where the bottom half of the ribosome is actually moving relative to the top half, and that facilitates the red and yellow molecules called tRNA as they move between the two binding sites. So what we try to learn is how specific interactions can actually govern these very large scale motions. In this particular rearrangement, what you'll see is a region that will appear in a circle here in a second. And what we're going to highlight is how those specific interactions between a few atoms can actually stall the entire process. And this can serve as something that helps facilitate more efficient and accurate expression of our genes. In another example, what we can look at is something like a viral capsid. Now in this simulation, this is a system that has 420 individual proteins that are all identical. And during maturation, where DNA is packaged in the viral capsid, the shell has to expand. And in this case, these tails, the 420 tails, also have to move about 30 angstroms from the inside of the capsid to the outside. So what you'll see here is roughly a 1 million atom simulation where the capsid spontaneously expands by about 30 to 40 angstroms, and all 1 million atoms have to move together. And what we can do is we can simulate this tens to hundreds of times and then talk about the statistics of maturation and try to identify what are the key elements or the key structural features that control these really essential biological processes. So thank you for your attention and hope you guys have a great meeting.